Okay, hello. It's now time to start. I'm here with Nelson Labs to present a talk on meeting new challenges to toxicological risk assessment, changes to ISO 10993-17 just came out in 2023. This is what happens after you do your chemistry testing that Steve just talked about a few minutes ago. You have to do a risk assessment on the chemicals for a submission to a regulator, FDA or otherwise. This has been a long time coming, the first update to the standard in only over 20 years, and they just published it 7 days ago, so this is kind of hot off the press. I was involved in that work group that wrote the thing and voted on it. Completely changes the methodology for what we do to assess the toxicity of the chemicals that are extracted from a medical device. The devices that were already approved and on the market under the old Part 17 risk assessment standard don't require a new risk assessment unless there's a change in the product as required by the biocompatibility guidance 10991. The key changes as I go through this so you don't get lost, it resolves several methodology problems and gaps that were in the old standard from 2002. And basically, one of them is a screening step to streamline large data sets to focus on compounds of greater toxicological concern. Another is approach to the exposure calculation, what the patient sees to correct an implausible mass balance flaw in the old toxicity evaluation. As you see on the left there, this is what happens when you extract a device. A lot comes out, but it comes out over time and depletes. In the old standard, you compared that as a estimated daily intake. Well, that's really overkill. The true daily intake in just a 24-hour increment is a small amount. Hence, they revised the standard. The box on the right here says they are now accounting for daily release and assessing the amount extracted, which is more realistic for risk assessment. 3 different ways, one using a generic toxicity threshold times an assumed number of release days. Another way is using a chemical-specific threshold, a microgram per kilogram per day, versus the total extracted from the device divided by an assumed release day. And the last is using a sequence of extractions every 24 hours, and you can compare the toxicity value in microgram per kilogram per day to the highest amount extracted from any one 24-hour period. There are also 2 methods for calculating a carcinogenic tolerable intake that weren't in the old standard, slope factor and TD50, and they're separate delineation of special uncertainty factors, which is how you make sure the device is really safe based on the extrapolation. The topics that I'm going to cover here are overview of the new process, the toxicological screening limit to reduce how many chemicals you have to evaluate, methodology for estimating the exposure dose to the patient from the chemistry data, methodology for application of a generic threshold of toxicological concern, deriving a tolerable intake from literature for non-cancer endpoints. Those are normative or mandatory requirements. Deriving a tolerable intake for the carcinogenic endpoint. Those are also normative. Using release kinetics, that's when you get your 24-hour series of extractions, to assess acute and subchronic exposure intervals along with relevant toxicity studies and also a couple of case studies. First, overview of the risk assessment process. The areas in red are the new topics where right up front at the top you can streamline and not have to evaluate some identified chemicals if they're below the screening. Next, when you derive a tolerable limit for a chemical, both the evaluation of how to derive it has changed and also how you estimate the dose to the patient has changed. And third, there's one not just for systemic endpoints, but also for contact irritants. I'll call the tolerable contact level, that's changed just slightly. And lastly, there's some uncertain areas that haven't been fully harmonized yet, like how you apply a generic TTC, a form of a screening threshold for unknowns. First, I'm going to go into the new toxicological screening limit. It reduces the number of chemicals to evaluate in the toxicological risk assessment. FDA estimates that about 15 to 38% fewer compounds will have to have a detailed evaluation of their toxicities. The TSL method has its bearings from ISO 21726 and ICHM 7 for thresholds of toxicological concern, But the new twist is that the TSL and the cumulative exposures are compared separately for each of the different contact duration categories of your device. So those categories are 0 to 1 day, 2 to 30 days, and greater than 30 days of patient exposure. The takeaways of this is it provides a rapid method to assess the total extracted amount. It might reduce the total effort and focuses on the chemicals found at more significant levels for more thorough evaluation. So it could result in a time saving. The scope and evaluation of the new screening limit has some limitations. You can't use it on unidentified compounds, which is kind of a shame, but at least it provides one tool. Irritation sensitization, it's mainly for systemic toxicity. It can't be used for cohorts of concern, certain highly toxic compounds specified in 21.726, and not for very young infants, less than 6 months old, or gas inhalation pathway exposures. They're based on a cumulative dose or a number of microgram per day allowed times the days of exposure, and they're based on mutagenic thresholds for every contact category spanning the clinical exposure period. The equation is TSL is the TTC, or screening threshold, times the least number of exposure days in that contact category. So as the red box in the center, for less than or equal to 30 days, the shortest possible exposure would just be 1 day. And you take the TTC in microgram per day times 1 day, and that gives you a comparison point of 120 micrograms. If your device is long-term, you also have to compare to the second category, which is a TTC of 20 microgram per day for longer term, times 30 days, which is rounded down from 31 being the shortest number of days in that longer clinical interval of 30 days to 1 year. So you have to evaluate all applicable contact duration categories for a given device. So at the bottom, if you have a long-term device, greater than 30 days exposure, you have to compare against the TSL of 120 and also against the TSL of 600. Essentially, it works out to be the 120 is the most stringent for the comparison. There are two examples of the use. I don't have a lot of time to cover these examples in detail, but you can just see how it applies by the highlighted areas here. A long-term device on the left, let's say you found 300 micrograms from the chemistry test, a single use of the device for long-term. You have to compare it against 120 and also against 600. It fails, so therefore you have to evaluate the individual chemical toxicity. The example on the right is a cumulative prolonged contact device, also detected 300 micrograms from your chemistry test. A new device, though, is used every day for up to 10 days. Essentially, because this is a cumulative exposure comparison metric, you have to multiply your 300 micrograms found in testing one device by 10 and compare that to the TSL of 120. And again, this indicates you can't screen it out. You have to go a full literature search of that chemical, that particular chemical. Then the second aspect is estimating the exposure to the patient from the chemistry data, and that's in Annex E. You calculate the worst case maximum exposure for each ICH-M7 and 10993-1 contact duration category for medical device that covers the clinical exposure category. So you might have to evaluate 0 to 1 day, 2 to 30 days, 31 to 365, and greater than 366 as separate evaluations for a chemical. When several compounds elicit the same harm at the same target organ, you're supposed to sum the exposure doses. And essentially there are two ways to do this in the standard. One is if you had just extracted everything from the device, the total quantity is then compared to, you multiply it by a scaling factor divided by the body weight, and then you divide by the least number of days in that interval. So if it's a 1 day interval, you divide it by 1. If it's a 30-day interval, you divide it by 30. And that's how you do the comparison. There's also a possibility that you had a more sophisticated chemistry test where you extracted in 24-hour increments representing 1 day increments of patient exposure. And in that case, the second equation, you take the highest quantity detected from any 24-hour time point from your chemistry results. And that's the value that you use to compare. It's multiplied by a scaling factor and divided by a body weight. Two examples of this method here for estimating exposure dose. On the left, it's a single-use device, repeat use every 30 days, or every 3 days, I'm sorry. And one device can be used at a time. So essentially, if you extracted 12,000 microgram, You divide that by the 60 kilogram adult body weight and divide by 1 day. That becomes your comparison for the 1 day exposure. And from 2 to 30 days, you take that amount again, 12,000 divided by 2 days, because that's the shortest possible time that the chemicals could be released from day 2 to day 30. It might all come off fast. And now, you would think, why don't we divide by 31 for the exposure period from 31 to 365? Well, in this case, you can repeat a new device every 3 days. And because of that, essentially at day 31, you might have just implanted a new device, and that might all be released in 2 days. So everything after the repeat interval gets used with the same number of release days as a minimum. The next device is more typical long-term implant tissue bone contact, up to 2 devices at a time. Maybe the largest device is 150 centimeters squared, and you tested a device of a smaller size. So you have to scale your data. Let's say you found 10,000 microgram. You multiply by 2 times 150 over 100, divided by 1 day, divided by 2 days, divided by 31 days. Those become the different amounts for the exposure intervals shown in the square in the right-hand side there for the estimated exposure dose for each period. Next, if you had release kinetics, those are the 24-hour sequences of extraction that are talked about in this standard. You essentially do a similar 4-part calculation, but you have time points of 24 hours to take advantage of that. On the left, externally communicating device, repeated use daily for 10 days. Up to 5 devices can be used at a time. Again, you have that scaling factor. Multiply the amount detected on the first 24-hour extraction times the scaling factor divided by 60 kilogram. That is the amount on extraction day 1. For the extraction days 2 to 10, because it can be used up to 10 days, you have 3 more extractions on day 2, on day 3, and day 4. Well, as you might expect, there's a depletion curve for the extractables coming off your medical device. 1,500 micrograms, then 500 micrograms, then 200 micrograms. For this, you have to take the highest or the worst case to assess your toxicity in that interval. And the one highlighted in blue there on day 2, 187, is the estimated exposure dose. The device on the right is another externally communicating device. One device is used up to 25 days. It's the same analogous concept. You used day 1, day 2, day 3 extractions. On day 4, it was not detected because it was lower. And again, you take the worst case amount of any of one of those extractions for that interval. Next, we're going to talk about what happens when you can't identify a compound, and it's called an unknown. Well, those are problematic for the FDA because you have to compare them to a very stringent generic threshold, assuming worst case toxicity. ISO 21726 and ICH M7 both specify these thresholds of toxicological concern called TTC down below. If your device only is used for 0 to 30 days, it's pretty lenient at 120 micrograms is the max for any unknown. If your device can be used from 1 month to 1 year, now it's 20 micrograms per day. So the longer it's used, all the way up to, if it's lifetime use, it's 1.5 microgram per day, a very stringent threshold. It's hard to see that low. And that's how you'd say an unknown is okay, even though we don't know what's in it. So this is used in 3 different ways. First, is part of that TSL approach. I just covered that for identified compounds. Second, for identified compounds when you can't find any toxicity data. Okay, so you can compare the EED max to the TTC. And third, assessing unidentified compounds, which is the vast majority of cases. And lastly, the same limitations apply that we just talked about for the TSL. There's special cohorts of concern in 21726 that you can't use it on. You can't use it for a device for very young infants. You have to assess toxicity of everything, and can't use it for gas inhalation pathway exposure devices. So a couple notes on that. Because this is the newest standard, it should harmonize with this TTC standard. It doesn't quite. First, there's no stipulation to adjust the TTC for body weight. So it doesn't matter if it's adult or child, you use the same TTC. Second, unknowns are excluded from the TSL. And so for an implant, that means your unknown can't be assessed using that magic comparison value of 120, which is easy to pass, you have to compare it to 1.5. That's hard to pass. Third, the methodology does not supersede the AET detection limit needs that are in the chemistry guideline part 18. Lastly, I'm going to say that 21.726 will be revised in the next work group 11 project, which I'll probably see in a few months when they get it off the ground, which might improve the harmonization. So we could see this change. Also, there's in silico methods, computer prediction of structure to establish toxicity. It was formerly used to establish a non-cancer TDC for compounds without tox data. It may be needed less often now because the old non-cancer TDC was actually more stringent than this new TSL. So we still might have to do it for high levels, but we don't do the computer prediction as often, and there's a rule-based computer prediction and a statistical base. Next, we're going to talk about deriving that tolerable intake for the non-cancer endpoints, everything except cancer, from Annex C. This is normative. The formula is familiar from the old standard. It equals a point of departure, usually a no effects level or a lowest effects level, divided by a bunch of uncertainty factors because you're extrapolating toxicity from an animal study to a human. And previously, the 2002 standard considered UF3, uncertainty factor 3, as a single consolidated factor for the net impact of all uncertainty sources besides interspecies and intraspecies. But now they require you to have separate uncertainty factors for each one. We'll go over some of those. And they have recommendations for what default values. Lastly, additivity is a new concept in here in 3 different places. When 2 or more constituents elicit the same harm at the same target organ or system and have the same tolerable intake, we must sum the estimated exposures for each constituent. Next, the margin of safety, which is, does it pass or fail? 2 or more constituents have to be summed when margins of safety have to be summed when they elicit the same harm in the same target organ, even if it's not the same chemical and the same mode of action applies. That's kind of a tricky little question there, requires some experience in toxicology. So the calculation there is, if you know the margin of safety, the index is greater than 1 is okay. That margin of safety from a sum total is equal to 1 over the sum of the margins of safety of the reciprocal of each individual chemical. Lastly, in some cases, you don't know a distinct mode of action. All you know is the liver weight changed or something like that, or minor change in blood chemistry that's reversible after the study. In those cases, you can say the risks are not additive. Here are those default values now that they've come up with that are different from before. Most commonly, an animal study of a chemical is an oral study. To extrapolate to a systemic exposure, you have to account that the bioavailability or the oral absorption wasn't 100%. They give you 2 options. One is a tiered approach, 100 if the bioavailability is really small, less than 1%, 10 if it's between 1% and 50% orally bioavailability, and 2 if it's between 50 to 90. An alternative approach, which is more nuanced, is you can take the no effect level and multiply it by the percent bioavailability, the percent absorption, which is more realistic, something that I pushed for to get it in there because it's often more realistic. Next, there's an uncertainty factor for exposure duration. So now if you had a 28-day study, subchronic, and you're trying to assess the longer term interval, 1 year or more, you have to use an extra uncertainty factor because toxicity may be worse after a longer time. And there's a default of 6 or a range of 2 to 6, and you have to justify that in your risk assessment. Similarly, subacute study applied to chronic, even worse. It's a greater extrapolation. The default is 10. Point of departure, if you don't know the no effects level, but a lowest effect is available, you have to pick a higher uncertainty. Lastly, data applicability. If you don't know the toxicity of your chemical, but there's a chemical structure of another chemical that's a surrogate or similar to it, you can apply that one and add another uncertainty factor. And lastly, data quality. If you're a non-GLP study, if you're familiar with biocompatibility, or if you're missing some details on what endpoints were evaluated by the pathologist, you have an extra uncertainty factor. Lastly, I'm going to show you that it really matters what toxicologists' experience level does this new standard. Because you have so many uncertainty factors, that makes these values much more stringent. And there are tools that you can use to assess this. I compared the 2002 and the 2023 guideline for a chemical. This is a typical carboxylic acid, a fatty acid. It forms a cyclic 5-membered ring under certain pH, and then in a different pH it opens up. We used a 28-day study for the entire assessment. Under the 2002 guideline, there's just one value. There's simply a value for extrapolation of oral to systemic, and we get an acceptable level. Now, under the new guideline, you're going to evaluate the one-day toxicity or acute separately from the chronic toxicity or the longer term, because we had to estimate those doses separately. Now we have two different values to compare to, and we get to use different uncertainty factors. This is where a toxicologist will use some expertise there where it will pass. So if we're extrapolating that 28-day study to a one-day exposure, there's no extra uncertainty because you've actually tested it longer. Whereas if you're extracting it from that 28-day to a long term, you need that extra uncertainty factor of 10. So hence, this is the manner in which you can pass. And it's also mitigated by the fact that the longer term exposure is divided by 31 days, the shortest interval longer than 30 days. And so that makes the exposure lower. So this is the way the two factors can work together. And calculation method requires a careful consideration of these uncertainties to develop a reasonably conservative allowable limit similar to the old guidelines. Next, we're going to have derivation of a non-cancer tolerable intake using a surrogate analog. I'm not going to go into all the details, but essentially people have been doing this for a while. Now it's codified in the standard. If you want to use a similar compound as a substitute for the toxicity of the compound lacking data, you consider chemical structure, physical chemical properties, oral bioavailability, and then you take into account the degree of differences between your chemical and the surrogate chemical with an uncertainty factor. And you also may need to consider the robustness. If you have a whole class of chemicals that are serving as a surrogate, you might have to derive more than one value and take the most stringent to make sure you have a robust toxicity estimate. Here are some examples, which I won't go into anything other than just to say their structures are quite similar. They would degrade in the same way, having the similar functional groups. Next is the carcinogenic endpoint toxicity value. If it's a known or suspected human carcinogen, this is now required. And there are now two methods in there. I actually worked on telling them what methods to use because of my environmental background years ago. That was a big part of environmental. The two types of carcinogenic endpoints are threshold and no threshold. So basically, most of the time, the default is there's no threshold. There's still some cancer risk the lower and lower and lower the level you're exposed to, but there is some risk. It's linearly proportional. And the concept for acceptance is less than 1 in 100,000 probability of developing cancer is considered acceptable. So there's two ways to do that. One is a slope factor. Usually a regulatory agency will publish a slope factor that's a potency so that the cancer risk is equal to the dose divided by the slope factor. You can rearrange that to solve for what is the dose that gives you a 1 × 10⁻⁵ risk, which is now the threshold limit. A modified approach, though, can be used if it's not lifetime exposure. Lifetime assumed to be 70 years times 365 days. If there's evidence that the release terminates leaching from the device after a specific length of time, then you can do a proportionality adjustment for the portion of a lifetime that represents. That's reference number 120 in there. And when you do it that way, down at the bottom, you can see cancer risk, specific dose, and added for emphasis is the subscript cumulative, cum, multiplied by 25,000 days in a lifetime divided by 1 day of exposure, if that same dose could be released in 1 day. And essentially what that means is the cancer risk from spread out over a lifetime for the same dose is the same as if you got all that dose all at once. Two examples, and again, I don't have time to go into a lot of detail, but the slope factor approach, we have two examples here. In the one case, it's a metal implant. So a metal implant won't really absorb chemicals strongly. Organic chemicals will just be on the surface as residues of the metal. And therefore, if you exhaustively extract the device, that means that the organic compounds would essentially be fully removed. And so whatever that total quantity is, you could treat that as the same risk as if it all came out in one day, because you know you're done and that's a lifetime exposure. So by doing it that way, on the bottom on the left there, it says 1 times 10 to the minus 5 divided by the slope factor times 25,000 over 1 day exposure. That's how you develop your acceptable level. On the right, you had a more sophisticated device where every 3 to 4 weeks, you put a new subcutaneous drug delivery product in the patient. And so the extraction took 7 days to release everything, but as a worst case, we assume it all can come out on the one use. But what happens though is that because you're using it every 3 to 4 weeks, that essentially means once every 21 days or 17 times a year is how many days you would be exposed to the entirety. So doing it that way, the cancer risk allowable limit becomes 1 times 10 to the minus 5 over the slope factor times 25,000 days divided by 17 days a year times 70 years. So again, it still gives you an adjustment. Next is a simpler method when somebody like EPA or California OEHAA didn't derive a slope factor, is a TD50. And that stands for the daily intake associated with 50% cancer incident in the animal study that was used. And so what you do is you take the TD50 divided by 50,000, and that becomes the dose at which you would get a 1 in 100,000 risk. A very simple concept, and you can do the same kind of rearrangement where if you know you have the whole release is equivalent to 1 day, you can use that same adjustment as on the preceding slide. Alternatively, you can do a linear extrapolation of what they call a benchmark dose. There might not be a 50% dose. The study might have derived a 10% cancer incidence called a BMDL10, and you can just proportion that 10% divided by that dose equals 10 to the minus 5 risk divided by the allowable dose at 1 in 100,000 risk. Lastly, the more sophisticated way is using this probability exponential equation, which all it is it shows that the cancer risk flattens out as you approach 100% chance of getting cancer as the dose goes up and up. And that exponential curve is what does that. You can solve that for the slope factor, and you can say if you had a 50% incident for the probability PI, you can solve that as TD50 is natural log over 2 divided by the slope factor. That's a little bit more conservative than doing it the other way. Lastly, intermittent to continuous dose extrapolation can be done with an animal study that wasn't dosed every day. So let's say you had doses, ICHM7 gives you an example. The daily dose was given to the animal 3 times a week, so you multiply by 3 over 7 to adjust that to an average daily dose, and then you input that into your toxicity calculation. Similarly, if it's less than 24 months exposure, which is lifetime for a rat, you can adjust it proportionally. Lastly, I gave 2 examples of the TD50 linear extrapolation, and due to time constraints, I am going to slow down now and just say these were both done using the TD50 over 50,000. And lastly, we have special cases that have to be justified called threshold carcinogens. A threshold carcinogen is something where below a certain level, the cancer risk drops off to 0. And those happen when it's not a mutagen, direct-acting mutagen. Those happen when it's caused by a series of reactions like inflammation, oxidative stress, modulation of receptor-mediated. And so ICHM7 recognizes that. US FDA M7 also recognizes it, and they say existence of mechanisms leading to a dose response that is nonlinear is increasingly recognized. So I'm just going to say that there's a way of doing that with a NOEL, which is a traditional way of doing it instead of extrapolating down to 10 to the minus 5. A NOEL divided by an extra uncertainty factor here of the ratio to 1% and an extra uncertainty factor of 10. So it's pretty conservative. It gets you down to about 0.1% equivalent level of exposure for cancer risk, but it's not as conservative as 10 to the minus 5. And I don't really have time to go through these, but one is for cobalt, which is in Europe, a restricted substance. And you can show that that's a threshold carcinogen by the influence of reactive oxygen species and other biological parameters. And you can derive it from a Lowell, and that has been accepted many times by Europe. And the example on the right is ICHM7 and aniline. They actually did that calculation for you and threw in an extra factor of 10 for severe toxicity, non-genotoxic carcinogenicity. Lastly, I don't have time to do this one, but the tolerable intake for release kinetics. Remember the study where you do every 24 hours extraction, okay? You got all those time points. Well, we covered the exposure. You're gonna sometimes be able to find different studies. You might have a subacute study that might only be 14 days or 28 days. And you can use that value. Hopefully it's a more lenient value. It's, because over a short time you can tolerate more. And you can use that for the tolerable intake for the 0 to 1 day result of the chemistry. And you can use a chronic study or a subchronic study for the longer exposure periods. And the one on the right is the same exact concept. One study was used for the subacute study for the 0 to 1 day, and a different study is used subchronic for the chronic TI. I have 2 case studies. Again, I'm just going to spend like 2 seconds on monomer elution study. That's an in situ curing polymer. It cures as it's in your body. So monomer, the reactants are still in your body when it's implanted. And it has a very nice kinetics curve. ASTM 451 has a method for doing that. Again, we did this before. Acute exposure, separate toxicity value, separate exposure estimate from the chronic. We did the same thing on that polymer for antibiotic doped polymer. It also has a depletion curve. The acute level was higher, and we evaluated an acute toxicity threshold against the 0 to 24 hour data and the chronic exposure threshold against the longer time points from extraction. What happens next? Regulatory agencies will review this standard. Remember, it just came out 7 days ago. When the review from each country or region is complete, they're going to issue a statement of recognition. It's possible that certain clauses in the standard might be excluded from recognition by some countries. Hint, hint, FDA. FDA never accepts everything without saying, no, we don't buy this. So we'll have to wait and see. During the transition period, regulators might accept the risk assessment done either way, the old standard or the new standard, but the intent to accept that is not a guarantee that we won't receive any comments or objections. So I just want to remind people of that, and that's pretty much it. Thank you very much. I went a couple minutes over, but I thought there was a lot of new stuff here. It's going to affect everything. The chemistry program that Steve just talked to you about in the last presentation, it's going to change how they do things, because now these requirements, how to use the chemistry, have changed. Any questions from the audience?
So the number of devices involved in the actual extraction, then you consider that number in your dose estimation, right?
That's correct. That was why the exposure estimate includes a scaling factor. Either if you use multiple devices in a patient, or if you use a larger device in a patient than the number that were extracted. This is how you take the result of the chemistry and multiply it by that scaling factor, so it does represent the clinical exposure, worst case, to the patient.
So, so my confusion here. So we used, I mean, in current practice, if you have the chemistry report that analyzes something per device, so microgram per device. Mm-hmm. So when you do with this new one, do we need to do any change or any modification from there? No. Or you just take it microgram per device?
Right. Your chemistry lab can give you the same data we used to get. Remember, the first one I showed was total quantity. You're extracting it either exhaustively until nothing else comes out, or for a short-term device, you might only need to do it for 72 hours. That's going to be assumed to be the total quantity released. You don't have to report that any different. You still take the same lab result. But what we do as a toxicologist is it's more realistic. I don't have to assume that amount happens every day as a patient exposure, because once it's all been leached, there should be no more to leach, right? So what we do now is we multiply by the shortest number of release days, and that is how we compare it to the benchmark.
Okay. So yeah, it's not changing on that perspective at all. Okay.
Now, if you also previously had simulated use or release kinetics in Part 18, they put that in the chemistry standard in 2020. And so if you get a high number from doing a total quantity extraction, you can always go back and test it with 24-hour extractions, and maybe you'll lower the temperature to body temperature. You might use milder solvent, but those 24-hour time points will certainly be less release than if you just kept extracting it until there's nothing left. And that provides a refined estimate for the toxicologist if the first pass wouldn't pass no matter every different avenue that they give you in the standard.
Thank you.
Okay. Any other questions? No? I stumped the panel. I probably went so far in the weeds that we just need to go back to this and relax your eyes a little bit. Incidentally, if you have any ISO questions, I'm on the committee for the risk assessment work group 11 and also for chemistry work group 14. So I'm involved in whenever they update one of those things. I get my 2 cents in, and everybody else does too, and we kind of vote on it and talk about, so I can often see what's coming down the pike before it happens. A lot of these changes were in the draft from 2021, okay? So. Yeah, that's a good question here. Going to the very last slide, how long does it take for a company or a country, whether it's FDA in the United States or Europe, to recognize a standard? And it's been getting better. In the old days, sometimes it would be a year or more. And lately, I've noticed in the last 3 years that most of the countries are recognizing things in 6 to 8 months now. So that would be my gut check, because I have contacts at FDA unofficially that have told me FDA is already on board. They were accepting some of these new risk assessments 2 months ago before it was final. And because they anticipated they were on board with most of the concepts. They have a seat at the table, to represent in developing these standards. And as a result, a lot of these guys were so tired of the old way of doing it. It's too simplistic. It wasn't realistic enough, that they have really put all their input. So I think FDA is going to move faster. I think we're going to see recognition in 6 months. So from now, that would be February or March.